So we are P4G2. My name is Malcolm Ma, 4.13.
My name is Jessica Barr, 4.2.
I am Samantha Shiganaga, 4.25.
I'm Carmen Payne, 4.20. So the gross anatomy of the bone consists of bones and ligaments, and muscles. Bones and muscles, and these muscles are indirectly attached through tendons and aponeuroses, or they are directly attached in the case of the periosteum fused with the epimysium.
And in the case of our presentation, we're doing attachment indirectly via a tendon. So in this case, the muscle will be attached to the bone by the tendon, and beneath the tendon, the muscle is wrapped in the epimysium. Within the epimysium, there are groups of fascicles, which are large groupings of muscle fibers. Each of those fascicles is going to be wrapped in the perimysium.
And each muscle fiber is wrapped in endomysium, and then each muscle fiber consists of myofibrils inside of it, and the sarcolemma surrounds it as the cell membrane.
Each myofibril is made of myofilaments, which are thick and thin. The thick filaments are myosin, and then the thin filaments are actin. So both of these myofilaments, these form the dark and light bands in the sarcomere, and these form the striations in the skeletal muscle.
So the dark bands are where the myosin and the actin overlap, and they are called A bands. Areas where there's only actin are lighter bands, and they are called the I band.
So within the A band, there is a middle zone called the H zone. In the H zone, around here, there should be an M line, which is the connection of the myosin. And when muscles contract, actin moves closer within the sarcomere, which contracts the entire sarcomere. And as that happens, the myosin heads will each attach to the binding sites on the actin. And this is the basic concept of muscle contraction.
Also, these green lines are the sarcomere boundaries, and they're formed by the connection of two actin filaments.
Those lines are also called the Z disc.
Yes.
And during contraction, the H zone shrinks, and the I bands do as well.
So a motor neuron sends a signal which releases neurotransmitters. In this case, it's called acetylcholine. The acetylcholine binds to the receptors on the sarcolemma, and after the acetylcholine binds with the receptors on the cell, an electrical impulse is sent, and then afterwards the ligand is bound to a enzyme which breaks it apart, so that no further signal is sent.
And this area here is called the synaptic cleft.
Okay, so the action potential is set along the sarcolemma and then down the T-tubule, and this triggers the purple, which represents the sarcoplasmic reticulum, to release the calcium. And then from there, so on the actin filament there is a low concentration of calcium, but once there is a higher concentration of calcium, the troponin moves the tropomyosin off the binding site, which then allows a myosin head to attach to it.
And then during contraction, the myosin head is attached to the actin, and the myosin head, ADP and phosphate are attached to the myosin head, and the myosin head pivots, and the ADP and phosphate are released, and the body sends ATP, and the myosin head detaches, and the ATP is hydrolyzed, and one phosphate is broken off of it, which leaves ADP and one phosphate, and the myosin head reattaches.
And this whole cycle of attachment where ATP is breaking off the heads, and these two are left, and then the head and the process is repeated over and over. It's cyclical, which happens until another signal is sent down the motor neuron to stop the contraction of the muscle, in which, when the muscle is relaxed, all of the actin filaments will sort of release and go back to where they originally were, and thus the sarcomere will then decompress and get larger.
Is that all? Yeah. Okay.